Mendus released Q1 report today, and we're discussing the report and upcoming events with CEO Erik Manting. Welcome, Erik.
Hey, Antti. Good to see you.
Erik, would you start by going through and summarizing the Q1? What were the main events and developments for Mendus?
Well, we were very happy that we made a lot of progress with our main partnerships today, which is a partnership with the Australasian Leukemia and Lymphoma Group, or ALLG, with whom we are planning a large trial, randomized controlled trial of our lead product, Vididencel, in combination with current standard of care, which is a drug called Onureg, or oral azacitidine. And we were very happy that they were with us in the business update in March, whereby we had anticipated ethics committee approval, which came in just a week later. So the week thereafter, we got the ethics committee approval, and we were able to start the trial. So we are currently in the startup modus of that trial. We have shipped product to Australia, and we are opening up the sites, etc. So we're very happy that we can push off this trial, which for us is a very important expansion of the clinical development of Vididencel. And the other partnership, which is extremely important for us, is a partnership with NorthX Biologics. They are a Sweden-based manufacturing organization for cell and gene therapy products. And the reason it's so important for us is that in order to be able to carry out a phase 3 and in the end also commercial launch, we need large-scale GMP manufacturing. We started working with NorthX after the summer of last year, and we were very happy in the business update we provided in the first quarter to also announce that the facility is now completed. So the construction of the manufacturing facility, we had started the first run, and you always need a number of runs to come in the end to a point where you can make GMP material. But the first runs, of course, are also very important when you actually start transferring the manufacturing process from Mendus as a non-GMP facility to NorthX in this now operational GMP facility. So that was for us an important moment in Q1. Another presentation, which was important for us, was around our NK cell program. As you may remember, we have developed a method for production of so-called memory NK cells. And memory NK cells are very interesting from a therapeutic perspective, but of course you need to be able to make therapeutic quantities. And that was the first step in the development of the program. And what we have now also added is initial data showing the efficacy of those NK cells in killing tumor cells. And that data we presented at the Innate Killer Summit, which is a leading conference in NK cell biology. And then finally, we raised additional money. We had warrants, so-called TO3 warrants, outstanding as part of last year's financing. And we were very happy to announce in the first quarter that all of our major shareholders, but also everybody in the management and the eligible board members, the ones that live outside of the US, participated in the conversion of their warrants. And we raised in the end around 69 million Swedish krona out of the conversion of those warrants. And that provides us with extra cash runway until the third quarter of next year.
You already mentioned your very important study Cadence that is about to start recruiting. So can you talk a little bit more about Cadence? What are the aims and goals and timelines of that study?
Yes, well, it's going to be a sizable trial. It's a trial run by ALLG, so of course in collaboration with us, and we make sure that all the study material, so the product is there to do the trial. We have lots of interactions on the scientific level. Next week, for example, is the Science Days, which is the days for all the investigators that work with ALLG, and our CMO, Jeroen Rovers, will also be there. So it's quite an intimate relationship, but in the end, it is their trial. So the only thing we can do, and they have also presented as part of our business update, but the most important hurdle we still had to take at that time was the ethics committee approval, which came in relatively shortly afterwards. So we're actually starting the trial as we speak. And that was the aim as a start. How exactly the recruitment of patients will go, first of all, we will not make announcements around it because it's ALLG's trial. But of course, we keep a close eye on how the trial is progressing. And what we do expect is initial safety data from the first patients treated in that trial. And what's very important for us also is that in parallel, we are preparing for a registration trial. that registration trial will be a company-sponsored trial, which we will carry out by ourselves or in combination with a partner. But one way or the other, we are making sure we are ready for such a trial in the second half of next year. And that trial will benefit from data from the CADENCE trial. So on the one hand, of course, it's good to already have some initial safety data in combination with ASA. We already had some initial safety data from the phase 1 trial we completed and also published end of last year, but it was completed already some time ago. Yeah, but there was some patients also treated with azacitidine. But this trial, the CADENCE trial, is a lot more structured in the way that we now really apply it with oral azacitidine as a maintenance drug. So the most important near-term milestone is those initial safety data, Antti. And then hopefully also, if that first cohort of patients goes well, we obviously can make a much bigger step into an additional 100 patients. And that's where we also start to expect to see a lot more significant clinical efficacy data.
You mentioned building readiness for the pivotal stage. So more specifically, what kind of milestones you need to achieve, kind of building up that capability?
Yes, it's actually a couple of milestones. The most important one is the preparation of the trial protocol. And of course, you have to think it through and you have to calculate it through and you have to talk about the trial protocol with the regulators, or the FDA in the US and the EMA in Europe, to make sure that the protocol that you in the end design provides for a good chance of your product to be approved in the end. The other element of the trial is making tactical choices about what kind of patient numbers, what kind of patient selection. We also alluded to it in the Q1 call. The CADENCE trial includes basically all AML patients that come out of high-dose chemo, meaning both patients that have been diagnosed with residual disease, so MRD positive patients, but also patients that have not been diagnosed with MRD. And the reason is that there's often false negatives. So also MRD negative patients, patients that are seemingly cured of their disease, still have a higher relapse rate. So the CADENCE trial is opening up a broader patient population, but for the phase 3, we may need to think differently. And for example, zoom in more on the high-risk patients, which are the patients with an MRD positive status, which is the patient population we also treated in the currently ongoing phase 2 trial. So that is the kind of preparations from a trial design. And then in parallel, we have the NorthX exercise, the alliance leading to large-scale manufacturing, which is also what you need to have in place before you are ready to engage in a phase 3 trial, because you don't ideally want to change your manufacturing process during such a registration trial. And also we have planned to use the initial GMP batches from NorthX to support that registration trial. So that's where the timeline comes together roughly mid next year.
In addition to the AML trial, you are studying Vididencel for ovarian cancer in the Allison trial. So would you go through the Allison trial? You are planning for a readout later this year. So what kind of data can we expect at that readout?
Well, first of all, it's the completion of the trial on the primary endpoints, which is safety and feasibility. Feasibility on the basis of, are we able to trigger immune responses against antigens that have been documented to be relevant for ovarian cancer? That's the current setup of the trial. We announced end of last year that the trial is fully recruited. So obviously also now with the vaccination schedule of Vididencel in the back of our mind, we have treated the last patients. So that is what will come together second half of this year. We have already indicated in earlier updates of the trial that the data look very promising. So we do see very broad and robust immune responses triggered by Vididencel in ovarian cancer patients against antigens that have in a lot of other studies before we started the phase 1, so published data from other clinical trials, for example, been shown to be relevant for ovarian cancer. Whether or not it will lead to survival benefit, that's of course a whole different story. So we will have to see how the phase 1 data do or do not point in a certain direction. I think it's fair to state that ovarian cancer is a very different disease from AML. AML is basically black and white. It's such aggressive disease that you either have AML or you don't have it. And when you have it, it's so quick that you are immediately in a what's called florid or full-blown relapse. So in the phase 1 in AML, we saw a black and white signal. We either saw patients with high disease burden not responding at all and very quickly relapsing and passing away afterwards. And then we saw a larger patient population that had a low disease burden. Not everybody was in a complete remission, but at least they had less than 10% blasts, so less than 10% disease-causing blood cells. And in that patient population, we could see that the durability of the response was very promising. It was 36 months median overall survival. So you start to see a very different picture, which we also saw in the phase 2. So either patients were very sick, or they had this durable remissions and overall survival in the end. Ovarian cancer is different disease. It's not so black and white. It's slower disease. Also, patients visit the hospital less regularly. So you can't always see what has happened in between. So we don't know yet at this point if we will pick up also an efficacy signal. But the primary readout will first be about all patients treated, all patients building up, or at least a majority of patients building up antigen-specific T-cell responses against relevant antigens, and safety. And then we have to see how we move forward, right? If we have a strong signal, it's an obvious thing to do. If we have a weak signal or some promising data, but not very decisive, we'll have to think if we want to spend time, money, and effort on that program versus the potential to broaden in hemato-oncology. So those are more tactical discussions we need to have after we know how the trial will read out.
Your second clinical stage product is called Ilixadencel, and you're investigating that for soft tissue sarcomas. So can you explain a little bit about why Ilixadencel is an attractive option for soft tissue sarcomas, and what are the latest news regarding your plans in that area?
Sure. With Ilixa, we had a couple of challenges. One was manufacturing, because we had to redesign the manufacturing process, which was successful. That was a precondition to move the product back into the clinic. The other part, and that is taking more time than we initially anticipated, is the repositioning of the product in the current immuno-oncology landscape. We had a significant amount of data in metastatic renal cell carcinoma, so kidney cancer, but those data were not competitive versus current standard of care, which is immune checkpoint inhibitors. And we took also some other decisions with respect to checkpoint inhibitors. We had an ongoing trial with Keytruda, one of the leading checkpoint inhibitors, which we stopped because we didn't see the effect we were looking for, which was responses in treatment-naive patients in combination with Ilixa and Pembro, so the Keytruda co-medication. So we had to rethink the way we position this product in the broader immuno-oncology landscape, which now has come to a more general conclusion that it is very unlikely that you will improve on the checkpoint inhibitors where they are effective. So we had to rethink our whole strategy, and we initially thought to go after gastrointestinal stromal tumors, with the simple reason being that they are very difficult to treat, and we had some initial promising data in GIST, in gastrointestinal stromal tumors. But it was a very narrow indication, and also we were in, let's say, partnership discussions around running a trial in GIST that we in the end were not able to complete end of last year. So we have now shifted to another setting, which is broader, the soft tissue sarcoma indication, of which GIST is a part, but soft tissue sarcomas is a broader group of sarcomas. So we've widened the net. We're having different discussions now, and we hope to conclude those in the first half of this year, so we can bring this product back into the clinic. And the reason we choose soft tissue sarcomas is that they are, first of all, addressable by the product, because we have to administer it into the tumor. But secondly, also because they don't respond to currently available checkpoint inhibitors. So that's a unique opportunity to see if we can turn these cold tumors, as they are called, more hot again. So to make them more immunogenic by administering Ilixadencel into those tumors. So that is how we ended up with soft tissue sarcomas.
You mentioned preclinical developments with the NK cells. I guess it's a little bit too early to talk about taking those to the clinical stage.
Well, most importantly is that we get to work with these cells. And there's also in the NK cell field quite a lot going on. People were particularly looking for better ways to make large quantities of these cells, and then started to modify the cells, like, for example, making NK CARs and what have you. So it's a relatively young field when you compare it to the T cell field. We have an interesting finding, because these memory NK cells that we are expanding have been associated with real clinical benefit. But it would be also fair to say that to develop this program into a clinical stage program will take a significant amount of time, which we have to balance, obviously, with our current clinical efforts. But it's certainly a program that we think is worth pursuing.
Erik, thank you very much for the interview and best of luck for the rest of the year.
Thanks so much, Antti. Speak soon.